Okay. Yes, I will start. So I'm going to be talking today about power analyses for genomic counts data. I think this was also titled as power analyses for RNAseq and microbiome data, which it is, but both of those are actually fall under sort of where the input is usually like a counts matrix. And so power analyses for them are pretty similar. So some preamble here. So actually all the slides and all the examples in more detail as their own Markdown notebooks are available at this GitHub link. And you can see that here. It's organized by RNAseq and Microbiome. And if you click on them, there's like a folder. It has all the data. It has an R Markdown notebook. And it has an HTML rendering. And then the microbiome one equivalently is in here as well. Additionally, if you navigate to ccv.jupiter.brown.edu, you are going to get a environment that it has all the packages needed to do the microbiome power analysis. It does not for the RNA-seq. So you won't be able to follow along with the RNA-seq one. But if you install the package on your like system or something, you'll be able to sort of do everything. And to follow along inside here, what you're gonna do is actually, I think the start screen looks like something like this. So you're gonna click on terminal, and then you're gonna click type in git clone. And you're gonna actually clone this repository. So it's git clone, and then you paste this. And then if you click this little refresh button here, or I guess it refreshes by itself, now you'll see there's this directory, power analyses. And if you click on it, you'll be able to see the same folders. And then, for example, if you click here, you'll be in this directory. And from here, if you click, what happens is if you click this plus tab, you can click this to start like a new R Markdown notebook. Well, it's actually a Jupyter notebook that has R. Okay. It's going to be a Jupyter notebook that has R, so you actually won't be loading this notebook itself, but what you can do is then copy over these code chunks. And when you paste them, you'll be running R code. So that's if you want to follow along later. And then I think the final thing I'll say is that there's also, if you sort of just want to read through the rendered notebooks, there is a blog post. I think it's on the GitHub repo link too, or in the README here. But there is a blog post that has the microbiome analysis already rendered. If you don't want to click any code, run any code, you can sort of just take a look here. And we're not going to do the RNA sequence since I don't have the package set up. But this is, maybe I'll just like scroll through it later, but it sort of looks like this. If you do have the packages, once you do have the packages installed, all the data is there. So you can sort of just like, when you go to the notebook, you just like execute it and everything will run. Cool. Okay. So let's talk about power, first of all. So in experiments, a lot of researchers are generally concerned with something called type 1 error, or alpha. This is usually what they're talking about when they're talking about p-value. Does anyone know what alpha is? Or what is type 1 error?
The probability of rejecting no when it's true.
Yeah, so So if this is like your null hypothesis and H1 is your alternative hypothesis, then yeah, alpha would be the probability that you reject the null hypothesis when it's true, or that basically you say there is an effect when there isn't an effect. So beta, or that is called the type 2 error, and it's kind of the opposite of that. So so say if the effect is there, beta then is the probability that you do not reject the effect even when it is there. So basically you don't see it even when it's there. So in a simple example, if your hypothesis is that your high expression of gene A is associated with a certain condition, and you have two groups, one with the condition and one without. If you have a power of 80%, then that's saying that 80% of the time, we're going to see gene A be differentially expressed between the two groups, and 20% we would not see. 20% of the time, we would not see this gene be differentially expressed between the two groups, even though it's actually there. So essentially type 2 error or beta is like false negatives, and alpha type 1 is false positives. So researchers tend to be concerned a lot more with type 1 error, but type 2 error is also very important to consider. So in terms of doing analysis, there's a question of why should you do power analyses? So the questions you generally want to answer is like, how many samples do I need to detect this effect with high probability? Or if you have like a limited budget and you only have this many samples, how much power would you have to detect the effect? And then the main reason that we often do power analyses with our collaborators is that grant proposals require them. Do people have ideas of other reasons you want to do power analyses?
When you're designing an experiment, you just want to know the sample size, or you want to do a follow-up if it turns out like the experiment might be underpowered, to see how many more follow-up samples you want to increase that power.
Yeah. If you want to, if your experiment is underpowered, then yeah, you might want to see how many samples you need in a follow-up study. Yeah, so I mean, first off, you probably don't want to get a false negative. You don't necessarily want to repeat your study. But also actually low power is tied to the non-reproducibility of studies. So the lower the power of a study is actually the lower the probability that the effect you observed is actually going to be considered statistically. The lower the probability that an observed effect that is considered to be statistically significant actually reflects a true effect. And then there's actually something called the winner's curse. So like, let's say your study is underpowered and you discover that there's an effect. What's actually probably happening is that due to like chance, like the effect there has to be a large effect size for you to discover it. And so then your estimate of the magnitude of the effect is going to be exaggerated. So if the true effect size is like 0.4, but the effect size you discover is like 0.6, that means that when you do subsequent studies, you're not going to find that effect if the effect is actually if you do subsequent studies with the same number of samples, then there's a good chance you're not going to see that effect again. And so first off, then that study is going to be non-reproducible. And then second off, you're going to have like an incorrect estimate of the effect size. Does that make sense? I guess this is summarized in the line. The effect inflation is worse for small, low-powered studies which can only detect effects that happen to be large. All right. So that's, I guess, in theory, why you should do these power studies. Besides for grant proposals, you want to have like a study that's actually can detect the effect that you're looking for and has like a correct estimate. In practice, power tends to be really low across many disciplines. This is in a bunch of psychology studies. The power in all of these studies was estimated to be 60% or less. In neuroscience studies, I gotta move this. There are many studies with power less than 11%. And actually there's like kind of a gap here, but yeah, there's a lot of studies here that have a power less than like 20% or 30%. And then this is psychology studies as well. And I think in these are different journals. And basically the estimated power in all of these studies was less than 80%. So most studies are underpowered in practice.
Is it expensive to hit that 80% power?
It is also, yes, it is also expensive to hit that 80% in practice for the most part.
Small effect size.
Yeah.
More expensive.
Yeah. Yeah. All right. Yeah, so it definitely gets more expensive to hit the recommended power depending on your effect size. Which is but it helps then to know how much power you have and what the ideal number of samples would be. So the next part we're actually going to get into actually doing a power analysis for microbiome data. But I'm going to briefly go over what counts data is. So for a lot of your like genomics experiments, you're going to be looking at counts matrices. And the idea behind these is like in your columns, you have samples, and then your rows tend to be features, whether they're like genes or taxa or something else. And then the values are the counts of the number of times a feature appears in a sample. So as a concrete example, this is the matrix for the RNA-seq analysis. But here the rows are different mouse genes. There are 4 columns here. There's 2 wild type innervated, which is like inside a muscle. There's 2 wild type innervated, which form one group. And then there's 2 wild type denervated, which forms another group. And then the values are the counts of the number of times this gene fragment appears in the sample. In terms of how to do power analyses for counts data, the number one thing you want to look for is counts matrices that will sort of like fit into your analysis that have this structure of like rows, columns, and counts. And then, so generally you provide the counts matrices, and then you either provide or estimate parameters that are part of the analysis. Examples of that are like expected fold change, sequencing depth, subsampling rate. And then usually whatever package you use is then going to simulate additional data sets based on on a specific like distribution of the counts with these parameters, or it will fit a model. For example, for RNA-seq, it will fit a negative binomial model to the data, and then it estimates like a power versus sample size ratio. In terms of finding and formatting data, the best data usually comes from a pilot study that you've done. But otherwise, if you find a study that's close enough, The factors that you want to consider are like the system, like mouse versus human, conditions. And then the thing that we usually consider is like the accessibility of accounts matrix. Like often with these studies, you get like raw reads, but they don't provide any intermediate files. You can always like run a pipeline yourself to go from the raw reads to the accounts matrix, but that's definitely extra work. And then for control data, there's a lot of databases out there. The microbiome data, you can, at least for human microbiome data, you can get it from a database like HMP. Okay. So we're gonna run through this analysis for microbiome studies. So as I said earlier, if you've cloned this into your Jupyter Hub, you should be able to go to, if you click on Power Analyses and then you click on Microbiome and Tutorial, you should be able to open this R Markdown notebook. That's kind of a text file here, but what we'll be doing is we'll just be copying segments over into a new Jupyter notebook that runs R. So in this tutorial, we're going to be using data from the Human Microbiome Project. And so in particular, we're going to be basically here, we find like shotgun metagenomics gut samples. And then we make some abundance tables. So we start by loading the packages that we need. So this is just, you can copy over and then paste here. Oops. Actually, I guess I could copy from here and paste. I'm going to set my path to data so that I can load all the data easily. So in this one, we've actually organized everything into accounts matrix. So if I copy this chunk and run it. Oh, okay. Okay. So this actually goes through how you get the data from HMP as well. So first, actually, they have like a project catalog where you can look at basically so HMP is the Human Microbiome Project. So you can look at this project catalog to look at basically like sequence reads that are associated with like a body site, a body subsite, the gene counts in the sequence read archive. And so basically you can just look at different sort of studies that are in there. And so if you're interested in something like the gut, what you would want to do is select from HMP. You're gonna select, you're basically gonna take the SRA IDs that are associated with a particular body site. In our case, our body site is called gastrointestinal tract because we're interested in the gut microbiome. And so you would be taking those columns and then from there, using the SRA ID to pull out, you would be pulling out the SRA IDs. And then here, this is saying that there are 147 samples that are from the gut microbiome. Let's see. Okay. So if I run this and then if I run this to pull everything out, and then I'd look at the length of the IDs, there are 147 samples. So with that list, with the list of SRA IDs, which I guess I can you can see what the SRA IDs look like too. Then you can you can pull them. You can download the abundance tables that are associated with all of them. They're actually in the GitHub repo, but if you did want to download them, there's the client that's called Aspera that you can use to do so. So once you have all of the abundance tables, you want to turn it into a counts matrix. So here I am reading in all of the abundance tables, and then I am computing. I am adding new columns, the number of reads, because they don't provide the number of reads in their abundance tables. So if I do this, I end up with, so my first file has basically bacterial samples, and then the counts for each bacteria in my sample, which is similar to what I want to look here. And then what I can do is join together all of the samples. So now you can see by joining together all of the samples, I have, well, I'll have to change the column names, but I have a table here where the rows are different bacterial species, and then the columns are different samples. And then finally, the values are different counts. And then this part is some cleanup to relabel the columns into the correct SRA IDs. So now I have my counts matrix for my microbiome data. So in the next step, I'm going to compute these what are called pairwise unweighted Jacquard distances, which are needed for sort of they're like population parameters for like within group distances in my matrix. And the reason we're computing the mean and the standard deviation is so that we can find an estimate of this parameter called the rarefaction, which is like sort of like a subsampling level. It matters for microbiome data, but it's like a parameter that you use. And so we're estimating sort of like what level of subsampling will give us OTU tables that give you the the population parameters, the mean and the standard deviation, close to this table that we have. That's actually like our control from the microbiome data. And this is taking a while. Okay. So here's the part where you would run. We actually like loaded these as objects, but here's the part where you would sort of run a bunch of like simulated tables at different levels of subsampling to see what gets you close to the mean and standard deviation that you find. And then you can basically plot the mean against the subsampling level to sort of find what subsampling level gives you the equivalent mean. And then similarly, there's another parameter that you need for this particular analysis. That's sort of the number of OTUs that will give you the standard deviation here, so you would run this command called #sd, which we've already run here. And so to save time, it's loaded as an R object. And then when you run it, you end up with this relationship, and then this is now log. These are log plots, but basically what level of OTU numbers gives you that standard deviation. Question. I have seen papers convert count data to relative abundances before calculating distance metrics, such as Bray-Curtis. Would you recommend using abundances or counts? Oh, it depends. For Bray-Curtis, I would use relative abundances. There are some models where you want the raw counts because they sort of account for. They try to account for. Oh no, no, I would use relative abundances actually. Because, yeah. Some methods, I think, if you like, for example, if you're using like DSCoV to do your analysis for microbiome data for some reason, then you would want the counts. But otherwise, I would use relative abundances. Okay. So both of these, so now you have an estimated subsampling level that gives you the same mean as your control data, and you have an estimated number of OTUs that gives you the same standard deviation as your control data, or something close anyway. And so now you have these parameters that you can provide and simulate power. And here, group size vector is sort of the number of samples that you would have in each group. We put 30 here. OTU number is the number you estimated from this #sd function. Rare death is the subsampling number you estimated from this #mean function. And then these other parameters are the sequence depth is what you expect your sequencing depth to be. And then the effect range, basically you can provide like a range of like what you think the effect size might be. And then this will basically give you, it will simulate power, and this will basically allow you to, function should be used for, okay. This will simulate sort of a list of OTU tables that you can then use to input into the your final power calculation. Here where this part is actually just checking that our mean and standard deviation of like the tables that we have of the OTU tables we simulate is close to what we computed above for using the Jacquard distance. Oh, okay. I killed my Jupyter lab. Let's see. I guess I could simulate less of them. Okay. MCL apply basically parallelizes your R, but I think we don't have 4 cores on here. On the Jupyter Hub at least, so I'm just converting it to lapply. Okay. So basically the mean and the standard deviation are still close enough. So now you can compute power with, so SP again gives you a bunch of OTU tables simulated with the specific sample size and different effect sizes. You can also simulate with different sample sizes and a specific effect size, or both. But here we just did for 30 samples with different effect sizes. Let's see if this part works. But here we are going to, this line computes power. Alpha here is your type 1 error, which we set at 0.05. And this is a small simulation. We're just going to do 10 bootstraps. But if you increase it, it will basically like bootstrap each distance matrix and sort of give you a better estimate of the power. So this computes your power for having 30 samples across these different effect sizes. here we're then subsetting to only look at oh, why did I look at those with power less than 0.95%? Okay, I don't know why I did this. But I subsetted I subsetted here to only look at those entries which return power less than 95% but greater than 20%. And then the rest of this is actually fitting a linear regression through my points. Let's see this plot. Okay, this looks different then. It only has 2 points. Well, the rest of this is actually fitting a linear regression through the points I have to sort of tell me the relationship between power and effect size. So you can see that as effect size increases, my power also increases. And as effect size gets smaller, power decreases as well. And you can plot something similar with like sample size to power. So at least in this tutorial, it turns out we have 80% power to detect an effect size of 0.01 and 90% power to detect an effect size of 0.017. I feel like a general question here is how do you know the effect size for your study? This paper that with the package that we use has actually a table that catalogs effect sizes with different distance measures. So here we use Jacquard, but yeah, if you use Bray-Curtis, you'd have a different effect size and also different comparisons that you can use as a point of reference. You can also, generally, you would use like the effect size that you have from like, that's like calculated in a different study with like the distances as well. And yeah, that's pretty much that microbiome power analysis tutorial. I'm not going to run the one for RNAseq, but I'll just go through it quickly. This one, it's the same idea. You want to have like a counts matrix. Here we pulled data from a different study. This is like mouse muscle. We get the matrix that we had before. If you do have like a list of like genes that you're interested in, you can input those. And then in terms of the power analysis for RNA-seq, this line tries to estimate the dispersion parameter for the negative binomial model. And then here we like provided parameters for that we sort of like guessed at, well, like what we expect the fold change to be, which is similar to like effect size. This one allows you to put in like a false detection rate as well. And of course, the number of samples. And then when you run this one, you can estimate the power for a given sample size. So if you have 5 samples, your power, at least with this data set, your power is 8%, which is very low. And with 6 samples, your power is 15%, which is still low. This package allows you to also like plot different power curves. So like as your like number of samples increases, if your like false detection rate changes, goes from like 0.01 to 0.05, or like your like expected fold change or your like effect size changes, like how much more power do you have? But yeah, that is like pretty much it for both of these kinds of analyses. You want to get your counts matrix somehow, you want to estimate your parameters, you plug them in, and then you get plots of sample size to power. Yeah. Does anyone have any questions?
Question.
Mhm.
You were saying that you have count data.
Yeah.
If you have different kinds of data, right, a different distribution, does the methodology to perform the power analysis change?
What do you mean different kinds of data?
So if you don't have count data, you just have measure of some variable. How does the power analysis change when your data is not no longer counted?
Yeah, oh yeah, if you just have like measurements or something, the one that's like commonly used in like yeah, where you have like measurements of like height or like some particular attribute, that usually you're like saying like, oh, I think this like follows like you'd be using like if you're using like a t-test or something to compare, you'd be looking at like means and then like comparing the means. And so there's like pretty standard statistical tests for those. And then a lot of people use this program called G-Power to estimate power when you have these more standardized tests. No, this is not the right page. What is it called? Oh no.
As you can see, like each of those, they're both the same kinds of count data, right? I mean, they're both count data, but rather than peak-analysis, you're assuming a negative number distribution, and you have kind of a totally different approach to analyzing that versus with microbiome data, where you're I don't know what is the team like a multinomial distribution behind the data?
Yeah.
Yeah, and it's more the models also used are different. In one case, you're using a GLM for RNAseq with microbiome. You're trying to use some sort of minimal kind of fixed on. But I think so I think it's going to depend on the on the model you assume works for your data set. But in general, all the commonality here is always that you want to understand what sort of sample sizes you in this cases. One is to understand sample size, but I think another thing which is also important is to sort of understand how do you define what your effect size. That becomes harder. Or it's not as straightforward as I don't know. More about that microbiome data. But sometimes I don't think say, oh yeah, I have protein that thing out.
Yeah, I guess the part I skipped over with the microbiome was the effect size is really actually really hard to estimate.
And how do you get those estimates? Because a lot of the power, you don't do post hoc power, you do your power analysis before you do your experiment. So how do you sort of get those values? I'm just putting numbers, I guess.
I mean, generally you look for like a study that's similar enough, and then if they don't provide, huh?
What if you're breaking new ground with this new study? And then you just need to do it a couple times to see how much.
Yeah, you do. I mean, you want to make sure. I mean, you want to choose reasonable numbers, right? I mean, so yes, if there is no previous study available, you can still figure out based on your model what parameters you're going to tweak and what those parameters represent in terms of, and then you construct an effect size based off of those parameters somehow, if you can.
I mean, it's like every study is basically novel. But like they'll have often there's something done in like a different system or like in like a slightly different condition or something. Yeah.
My question is both management power now and the RNA-seq, I noticed that in all the other parameter to communicate on is the sequencing depth, and that's always been kind of mysterious for me. Is it just like, you look at, say, oh, is it Illumina or nanopore, and you say what species it is, and you kind of just, go from there, like what the sequencing depth is, or, yeah, just trying to understand how we get that variable.
Oh, well, your sequencing depth is like pretty much dependent on your budget usually. I mean, and usually people are like, oh, I want to sequence to like 30,000 reads or something. There's like, well, a question that comes up a lot is like, can we increase sequencing depth instead of increasing sample size? Because it's much cheaper to sequence deeper than to collect more samples. But there's I think at least 2 or 3 papers showing that like sequencing depth only increases your power up to like a certain point, like 30K reads or something per sample.
30 million.
Sorry. Yes. Wow. That's what I was like, wait, something sounds wrong. 30 million reads per sample. And then after that, your power, like it doesn't change at all. So you need more samples. Yeah. Oh yeah, I think I did that in the notebook because I was asked to see if they could increase sequencing depth.
But obviously, one sequence deep enough that there's no more effect to be determined at that point.
Thanks.